Record. Okay, so as usual, the typical announcements. I've extended quiz 7 and 8 to 11:59 p.m. tomorrow. If you haven't done so already, please do it. Quiz 9 will be out tonight. And if you have not uploaded your presentations already, please upload them because it won't let me put the grade in if you don't upload it, okay? So we are gonna do presentations, which let me just get out of this share. And who wants to go first?
Can I go?
Sure.
Okay. Can you guys see my screen?
Yes.
Okay. Okay, so the topic that I got was transcription and genetic code. The question I got was number 3, and it was, how was it demonstrated that the genetic code consists of triplet nucleotides? The options were A, RNA always contains a number of nucleotides that is a multiple of 3. B, all of the other answers are correct. C, an insertion or deletion of 3 nucleotides, but not of 1 or 2 nucleotides within a coding region of a gene preserves the function of the resulting protein. D, an RNA that is 300 nucleotides long always codes for 100 different amino acids. And E, every codon codes for 3 different amino acids, 1 for each nucleotide comprising the codon. So the answer that I got was C, an insertion or deletion of 3 nucleotides, but not of 1 or 2 nucleotides within a coding region of a gene preserves the function of the resulting protein. I said this because it is demonstrated that the genetic code consists of triplet nucleotides, because the genetic code is a triplet nucleotide, which means that 3 nucleotides are necessary to code for 1 amino acid. So if 1 or 2 nucleotides are inserted into a protein coding region, it can cause a frame shift mutation, which would change the amino acid sequence. And furthermore, so I have a picture over here to the right, and it basically shows the frame shift mutations. So if 1 or 2 base insertions shifts the reading frame of codons, it results in a non-functional protein. And if 1 or 2 base deletion shifts the reading frame of codons, it results in a non-functional protein. And we can conclude that from this that 3 base deletions or insertion restores the reading frame of codons, which results in a functional protein. So from this, we can see that the insertion or deletion of 3 nucleotides resulted in the preservation of the protein. So C would be the correct answer.
Good.
And that's it. Okay.
Anybody have any questions? We cool with this question?
And I uploaded the PowerPoint.
Okay, yeah, great. Okay, who wants to go next?
I'm supposed to go, but I just realized that I did the wrong question. I actually just did his question. So can I fix that real quick and then present at the end of the session, maybe?
Okay.
Is that fine?
If anything, you can present next week as well, if anything.
Okay, I'll just work on it quick. It shouldn't take long. I apologize. I just can't read. I'm sorry.
That's okay.
I wasn't here last week, so I can do mine this week.
Okay.
All right, so my question was the first question of the chromosomal mutations problem. So the question was, for a species with a diploid number of 60, indicate how many chromosomes will be present in each of the following somatic nuclei. So for the haploid cell, it would be 30 because the haploid number is half of the diploid number, which is 60. For the tetraploid, it would be 120 because it's 4 times the haploid number. For trisomic cells, they have 61 chromosomes because you take 2n, which is the diploid number plus 1. And then for monosomic cells, it's 2n minus 1, so you get 59. So the answers are haploid 30, tetraploid 120, trisomic 61, and monosomic 59.
Good. Everyone cool with this question? With the different definitions of and terminologies?
Honestly, I didn't understand it, but I can read over it again.
Okay, where did we lose you, I guess?
I don't know. When you do math, I can't follow math fast, so you have to speak to me like I'm 5, I think.
Okay. We can go over it again if you want.
I would appreciate it.
Okay. Am I going over it?
Up
No,
to
I'm
you.
in the class. All right. So for the haploid cell, it's half of 60 because the haploid means that there's one set of each chromosome and the diploid would be 2 of them. And then for the tetraploid, you take the haploid number, which is 30, and you multiply by 4 to get 120.
Okay, that makes sense. All right, no problem.
All right.
Okay. Let me just pull up something. Okay. Ariana, are you still presenting today?
Yeah, I have mine ready.
Okay. Share it when you're ready.
Okay. Right. All right, so I have question number 1, and it's, what is an interior triplet in both prokaryotes and eukaryotes? And what amino acid is recruited by this triplet? I chose answer D, AUG and methionine, because UAG, UAA, and AGU are all stop codons, while AUG is the only initiator codon here, which would narrow the answers down to C and D. The only amino acid recruited by AUG is methionine, which is the first amino acid incorporated into any new protein. You can see over here that AUG is an initiator, and then methionine is paired with it.
Good. Does that make sense? Are we cool with that question?
Yeah.
Yeah.
Wonderful. Okay. Let's go back to my presentation. Okay. So, okay. If technology likes me today, that will help. Okay. So this is another question I pulled out from our textbook. The mRNA formed from repeating tetranucleotide UUAC incorporates only 3 amino acids, but the use of UAUC incorporates 4 amino acids. Why is this occurring? Do you guys have any ideas? It's okay if you don't. Just gotta tell me.
I have no idea.
Okay. Good to know. Okay, so we have a repeating sequence, right, for both of these. So when you have a repeating sequence of UUAC, you end up with having leucine for UUA, and then hold on. Okay. And then tyrosine for one set, and then ACU ends up with threonine, and then you have leucine with CUU, right? So it gives you three different amino acids. And why is this happening? Because the code itself is degenerate in the sense that multiple codons can end up coding for another. Okay, let me reword that. It is degenerate in that it is multiple codons can code for the same amino acid, right? So that's the reason why this is happening that way. And then we have this sequence, this multiple one guy. We have tyrosine, isoleucine, serine, and leucine, right? So in this case, the degeneracy is not revealed because all of the codons end up producing different amino acids. Does that make sense? Did I completely lose everyone?
No, I kind of understand it.
Okay, good. So essentially it is because of the degeneracy of the codons. We cool with this question?
Yes.
Okay. Okay, so next we're gonna go into a little bit of concepts because the next homework that is due involves transcription and translation, and I want to make sure that you guys have enough of the concepts to get through that homework. This week will be mostly concept side, but next week we'll cover more on questions and be like knocking them all down, okay? So first, we're going to talk about the gene structure. So with prokaryotes, you have a promoter, you have the upstream end of the gene, which is at the 5' end. You have the promoter, and then you have a start site for transcription. And then you have a RNA coding region, or the coding region of the gene. And then you have this area where it's called the terminator. So this is where that ends with transcription, okay? And then you have the 3' end where that is downstream of the gene, okay? Then you have the eukaryotic gene structure. With this guy, we have enhancers that are upstream of the gene, and then you have some proximal control elements depending on what is needed, and then you have your promoter. Your promoter is the one that's like, okay, we need to turn this on to make sure we have this mRNA, that kind of thing, okay? So that promoter is there. Then you have the exons and the introns. So in prokaryotes, no introns. Eukaryotes, there are introns. And with this, you end up going in. One of the very last exon has a sequence where it's coding for a poly A signal, which is part of RNA processing, which we will cover too. And then you have a termination region. So that's essentially the structure for both of the types of genes, okay? So with transcription, you have so transcription is essentially making RNA, okay? So in prokaryotes and eukaryotes, there are differences in how transcription occurs, but they occur with the same 3 steps: initiation, elongation, and termination, okay? So in prokaryotes, it starts off with the whole core polymerase. Without this guy, you will not have anything happen. So this guy here has 2 alpha units, a beta unit, a beta prime unit, and an omega unit. Okay, so it's made up of 5 different subunits. And in initiation, this core polymerase binds to what is called a sigma factor. And this allows initiation to kind of like begin. So you're initiating the process of transcription, and it recognizes the different sequences that is required for transcription. So once that region is found, you have the promoter found. It's like, great, okay. Now the sigma factor is like, okay, core polymerase, go on doing what you're doing. And the core enzyme or the holoenzyme ends up, the core enzyme itself just goes on transcribing until it reaches a stop codon, right? Or a termination point, okay? Does that make sense so far?
Yeah.
Okay.
Yeah.
Good. Because next, oh, okay. So this is just essentially a question where you're just drawing it out. So essentially you have your DNA. We have our components of a prokaryotic gene. When it's transcribed, you end up with a 5' UTR and a 3' UTR and your coded region. It's all happy and it's ready to be translated. But before we go into that, we're going to talk about eukaryotic transcription. So with eukaryotic transcription, you have a Tata box. That is part of your DNA, okay? So we have this little box. It's like, okay, people, we need to start initiating all the steps for transcription. So it's like, come on, people, come on. And the TBP or Tata box binding protein and other transcription factors come in, and that forms transcription factor D. That comes in, binds to it, and it starts bringing in all the other components like transcription factor 2B, and then the polymerases come in. So there are 3 types of polymerases. RNA polymerase 1. So these are RNA polymerases. There are DNA polymerases as well, but we're going to focus on the RNA polymerases right now. So the first RNA polymerase, or RNA polymerase 1, those guys deal with ribosomal RNA, okay? So they form anything that looks like ribosomal RNA. Then you have your RNA polymerase 2. Those are the guys that help create our messenger RNAs. And then we have our RNA polymerase 3, which is helping with transfer RNAs, okay? Ooh. Okay, so we have this, so we have that. So in this case, since we're coding for proteins and all that, RNA polymerase 2 comes in, right? It comes in and it brings its buddy transcription factor 2F with it, okay? And then it's like, okay, all of these comes in, and then more of the transcription factors come in. And RNA polymerase, just once these guys are in, these transcription factors are in, they're like woohoo, yeah, it's go time, people. We need to get some mRNAs out, right? So RNA polymerase 2 continues until it recognizes a region where it terminates that process. And then it goes into that section where it polyadenylates, right? So it sees that sequence where it's like polyadenylation, and it's like, okay, we are capped. We have our poly A tail, and that mRNA is happy. Does that make sense?
Yes.
Okay. I know this is a lot of information, so if I'm going too fast or anything, stop me. Okay. So now we have our pre-mRNA, right? Because in eukaryotes, you need RNA processing because you have introns there, and it's like, hey, they don't code for any of the proteins that exist. So it's like, what do we do? What is next? So the next process that they do is RNA processing. RNA processing involves the methyl cap, splicing, and the polyadenylation. So that's part of that, okay? So with prokaryotes, transcription and translation occurs simultaneously in the cytoplasm. And this is because since we don't have any introns, it can directly happen simultaneously because you don't have to process the RNA. Whereas with eukaryotes, transcription and RNA processing occur in the nucleus and translation occurs in the cytoplasm. So since we have a lot of introns going on, to keep that flow going, the RNA has to be processed before it even enters the cytoplasm. Does that make sense? Okay. So again, this is another question that is kind of similar to how we drew our prokaryotic gene. So again, we have our DNA. It got transcribed, and you have your transcription start site here. You have your exon, intron, so forth. And then you have that termination site with the polyadenylation signal, okay? So in this case, they add on that cap that they need. So this is the 5' cap. Then the 5' UTR, and then you have all of the exons spliced together, and it's shortened down to what we need. And then you add on a poly A tail after the 3' UTR. Does that make sense?
Yeah, I think.
Okay. If I completely lose you guys, let me know. Okay. But it's,
This is a lot of information, but I think I'm getting it.
yeah, I know it's a lot of information. It's like 2 lecture slides crammed into one. So that's why I'm kind of going more on concepts today, just because if you guys don't know this, the homework's going to be a little bit more complicated. But any questions so far? Because in about a few slides, we're going to go into translation. So are we okay with this stuff so far?
Yeah.
Okay. If any questions come up, just be like, Mimi, hold on, what's going on? Okay. So in which cellular organelle do the 3 post-transcriptional modifications often seen in the maturation of mRNA in eukaryotes occur? So, ooh, something in the chat. Yep. Okay. Yes. Nucleus, right? So post-transcriptional modifications essentially is another way to say RNA processing. Oh, no. So that is in the nucleus. If they asked prokaryotes, they don't do this that much because the splicing part doesn't exist. So it's like but anything that is post-transcriptional in prokaryotes, that will be ending up in cytoplasm. Does that make sense?
Yeah.
Okay, good. Okay, so alternative splicing here is just essentially saying that there are different ways to splice the mRNA that you get. So you have your pre-mRNA, right? So this can go either way depending on the path or what signals they get to actually get the desired protein that they need. So in this case, the first case is like you want this protein. So they're like red, yellow, blue. And then that goes in. It's like, okay, so it skips the green and be like, here's your protein. But then another signal comes in and be like, we want red, green, and blue. So we take red, green, and blue, and then it splices it out, and it's like, okay, now it's come together. And then they form that protein. Does that make sense with alternative splicing? It's like the most basic version that I could find of this guy. Does that make sense? It's just how they end up splicing, where they end up doing that between the different exons. Okay, I'm gonna think we're cool. So, any questions before we go into translation?
Yeah, I just have a quick question.
What's up?
So like, I don't know what I don't know about the splicing, but can you just like explain it again? Like,
Okay,
I just don't understand what I don't understand, you know?
Okay, let's go back to this guy. So here we see the introns, right? So these introns don't actually code for the proteins that we need to get out of this. So the way so these ends of the exons have a specific site where it's called splicing sites. Those sites end up being, okay, so these kind of come in, they're like, okay, we need to splice it out so that this exon sites, they have splicing sites. Depending on the signal that the promoter receives, they end up so they're taking out the introns. But in the case of alternative splicing, you have multiple exons in here. But depending on the isoform that you need or the protein that you need for that specific process, then it starts jumping around between the exons. In some cases, a certain exon is not required for a certain protein that the cell needs. So that's when they come in and they're like, okay, we splice it out, we take this guy out, we're like, this guy comes here and attaches it to itself. Does that make sense?
Yeah.
Yeah, I think so.
Okay. It's essentially different ways of like combining different exons to form different types of proteins. Maybe it'll make more sense when we do. So essentially, splicing is just a way to get rid of introns, right? We don't want any any DNA that is not coding for our proteins, right? But in this case, sometimes different protein, so the same gene can code for different proteins. When this happens, sometimes you need to skip an exon or two, depending on how that protein needs to be formed. Does that clear it up a little bit more?
Yeah, I think I get it.
Okay, good, good, okay. Everyone else okay?
Yeah.
Yeah.
Great. Okay, I'm happy now. Okay, so now we're gonna go into translation. Translation is essentially a process of converting the information in mRNA molecule into a sequence of amino acids that make up the protein. So amino acids are the building blocks to protein, and translation is just the process of how you would do that, okay? So translation requires ribosomes, transfer RNAs, amino acids, and a lot of energy, okay? So this is just a little diagram here to kind of visualize the steps that it goes of the central dogma. Okay. So what does a ribosome do? We have the ribosome. It actually provides the scaffolding as to where the transfer RNA and the mRNA are brought together and allows some enzymatic activity to form peptide bonds, because you need to connect the two peptides, right? The amino acids, because if you don't have that, you can't make a protein. So in prokaryotes, you have a ribosome that is called the 70S ribosome. This 70S ribosome has two components called 30S subunit, which is the small subunit, and then you have the 50S subunit, which is the large subunit. This is for prokaryotic ribosomes. Then we have eukaryotic ribosomes, which is called the 80S ribosome. The small subunit here is 40S, the large one is 60S. Okay? And it has its own miniature sized ribosomal RNAs going on. Okay? So to decode and all this, tRNAs are involved. So tRNAs, they have a specific amino acid covalently attached to the 3' end of the tRNA. And it comes down to binding the mRNA at the codon. So remember codons, they're the 3 nucleotide pairs, triplet code, and then it actually folds into a cloverleaf shape, which is its secondary structure. And the anticodon loop down here is where it has the complementary strand to the mRNA codon. Does that make sense? Just essentially the structure of transfer RNAs and how they kind of come into play. Okay. So Again, translation occurs in 3 different steps like transcription, initiation, elongation, and termination. So in initiation with prokaryotes, you have an initiation complex. You have the small ribosomal subunit, initiation factors, and mRNA at the codon AUG. So our start codon. And it combines with the large ribosomal unit. And this sequence called the Shine-Dalgarno sequence, this sequence is right before the AUG start codon, right? So this codon here, so this sequence here is a couple of base pairs ahead of the AUG start codon. And this allows it to base pair with it in regions of the 16S ribosomal RNA in the 30S subunit, right? So this is how it gets initiated in prokaryotes, okay? Next with eukaryotes, so remember the 3 parts of RNA processing involves a 5' cap, right? So this is the name of that cap. So it's a 7-methylguanosine cap or 5' cap. That cap is how the ribosomal subunit 40S ends up recognizing it. So it's like a recognition sequence like the Shine-Dalgarno sequence in prokaryotes. And it's like, okay, now it's there. It's gonna go until it sees a start codon, right? And then that's how it ends up forming the initiation complex. Does that make sense so far?
Yes, this makes sense.
Okay, good. So when the when it comes to AUG, it comes down. It's like, okay, we brought in our methionine. Okay. So in elongation, this is where the different parts of the ribosome comes in. Okay. Okay. So the tRNAs bring the amino acids one by one, adding on to the polypeptide chain. So this is in the step of elongation. In elongation, the ribosome itself has 3 sites. One is the amino acyl site, which is essentially bringing in the charged amino acyl tRNAs. So all the tRNAs are coming in. It has a little amino acid attached to its 3' end, and it's like, okay, what do we do next, right? Then it goes into the peptidyl site or the P site. That ends up being the next charged tRNA to actually bind, start forming peptide bonds. So in this case, when you think peptidyl site, think peptide bond. That's what's occurring. You're making peptide bonds at the P site. And then the next one, so in this case, the protein polypeptide chain is still growing, but it hasn't dissociated yet. Then that's when the exit site comes in, or the E site. This actually releases the dissociated tRNAs so that they can actually be recharged with a free amino acid and then kind of come back into the cycle of making sure that you have the right polypeptide, that the amino acid that you need. Does that make sense?
Yeah.
Okay. So next and finally, we have what's called termination in translation. This occurs when you see a stop codon. So one of the three stop codons come in and you have no complementary tRNA. So instead of a tRNA, a release factor comes in and binds to that stop codon and then stalls the ribosome long enough to release that tRNA and the polypeptide. Does that make sense?
What do you mean by stalls?
So essentially, like in the sense, you know how the ribosome is moving through the mRNA, right? So it has to move through the mRNA to get it, to read it in a sense. Does that make sense? So
Yeah.
it's like stopping the ribosome from continuing any further, because if it continues any further, it's just gonna have not much of like actual amino acids that would be relevant to your protein. So it stops the ribosome and then releases everything else.
So it just permanently stops it or pauses it?
It pauses it in the sense of this one. And then if another mRNA comes in, so essentially here it's separating it out until the next mRNA comes in and does the whole initiation process.
Okay, that makes sense.
Okay, good. One more slide and I'll let you guys have a break, I guess. So essentially, then after it's released, you have your protein, right? You have your primary structure of your protein. This protein, the primary structure is the amino acid sequence. So the structure of it depends on the sequence itself, right? So once it comes in together, it's like, okay, it changes on how it wants to be formed. Sometimes it could be an alpha helix or a beta helix. Those are called secondary structures. Then all of these different structures come in, they kind of orient themselves and form a tertiary structure. And then it ends up with a quaternary structure with a complex of separate different little molecules of protein. Are we okay?
Yeah, that's pretty simple.
Okay. So I know I put a lot of information in this one, but any questions, concerns? Did I freak you all out with the amount of information?
Yeah, it just kind of seems like a lot. I'm gonna have to go over it a few times.
Yeah,
I will too.
trust me, I know. I know it's a lot of information because I was like doing this and I was like, because like I plan using the mastering genetics homework and it was combining both of these guys and I was like, oh boy, this is going to be a fun assignment. So I was like, okay, let me make sure you guys have at least something to go on. But trust me, we will go over more stuff next week on this. So I'm gonna let that soak in to you guys, I guess.
I do have a question about this material so far. It doesn't have that much complex math involved. I was wondering if the next exam will be like the last exam where it had a ton of different types of math I have to do.
With these topics, I'm not entirely sure. Typically, with like transcription, translation, you don't see that much math, but I'm not entirely sure. I don't really see the exam beforehand until you guys actually take it. So,
Okay. All right.
yeah. Caleb, did you still wanted to do your presentation? I know.
Yeah, I can do it. That's fine.
Okay.
Am I good to share now?
You should be able to.
Oops. Okay. Come on. Oh, so my question was on transcription and the genetic code. My question was number 2. Which of the following statements are false about eukaryotic mRNA? It was RNA processing involves 5' capping and 3' capping polydenylation and RNA splicing, or B, processing of nascent mRNA occurs in the nucleus. C, pre-RNA contains introns and exons. D, transcription and translation occurs simultaneously, or none of the above. So the first one can't be right because it is true. RNA processing does involve 5' and 3' ends. B, again, is true, so it's not the right answer. Processing of nascent mRNA occurs. It does occur in the nucleus, and that's just where the processing does occur. C, pre-RNA contains introns and exons. That is also true. Pre-RNA contains those coding and non-coding areas. And it can't be E, obviously, because D is the correct answer. Oh, big check mark.
I like the check mark. But any questions on this question?
No.
Cool. Good work, Caleb.
I already submitted the wrong question, so do you want me to resubmit this PowerPoint?
Yes. I will clear that attempt a little later.
Okay, perfect. Thank you.
Okay. Any questions, concerns?
No.
No.
Did I scare you guys away with all this information?
No, but I'm curious, what chapter is the ribosome stuff? Is it 13?
So I think ribosomes are in chapter 14.
Okay.
But let me just double check. That way I don't give you false information. And then you guys are going to be like, what the hell is Nimi talking about? Yep. So chapter 14.
Awesome. Thank you.
Well, if you guys don't have any other questions, you are free to leave.
Thank
Okay,
you.
thank you.
Yep.
Thank you. Have a good night.
You too. You guys okay?